So what I told, in each replicated structure, one half, that is old one half is of parental structure and one half new. Okay, and one half is of daughter structure. This is known as semi conservative replication. Okay, so this is known as what? Semi conservative replication. Okay, now one scientist were there, that name is Watson and Crick. So he suggested DNA replication is semi conservative in 1953. What he told? Watson and Crick suggested in 1953 that DNA replication is semi conservative in nature. Okay, which scientists were there? Watson and Crick. He told, or he suggested, that DNA replication is semi conservative in nature. Okay, now one more scientist were there. That scientist name is Taylor, and he suggested that replication. Okay, that we will see in a replication. Okay, so first we have to come on that how this replication occurs or transcription occurs or translation occurs. But before going into that, we have to see packaging of DNA helix. Okay, so come on that. Packaging of DNA helix. Okay, now why this packaging needed? Okay, so first we have to see, already we know that the distance between two consecutive base pair we have seen in double helix model. Okay, so what was that? 0.34 nanometer. So come on that, then we will see the distance between two consecutive base pair. Okay, the distance between two consecutive base pair, what we have seen is 0.34 nanometer or 3.4 angstrom. If we are converting this word in meter, so 0.34 into 10 to the power minus 9 meter. What we can say? We can write that one. Now, we also seen that the length of a haploid cell, okay, is 6.6 into 10 to the power 9. Okay, haploid 3.3, but diploid it will come double. So 6.6 into 10 to the power 9 and into 3.34 into 10 to minus 9 meters. So how it will come? 2.2 meter. What I told, you have to see that. We already studied the distance, the length of DNA from a diploid cell we have to see. We have seen the length of DNA from a diploid cell. Okay, haploid we seen 3.3 into 10 to the power of 4, 10 to the power 6. Okay, base pair. So we have to see is equal to 3.3 into 10 to the power 9 base pair. But here it is asking diploid. It is for haploid. Starting in the start class, okay, in the initial classes we have seen base pair is in haploid cell 3.3 into 10 to the power 9. But here asking diploid cell, so into 2. So what will come here? 6.6 into 10 to the power 9 base pair. Now 6.6 into 10 to the power 9, and per base pair recently we have seen 0.34 into 10 to the power minus 9 meter. Okay, so this 10 to the power 9 and 10 to the minus 9 will out. So what will come if we are multiplying this? So this length is 2.2 meter. This is the length of DNA from a diploid cell. Now, if we are seeing that size of typical nucleus, so what is that? 10 to the power minus 6 meter. Now, come on that. The size of typical nucleus is equal to 10 to the power minus 6 meter. So the size of nucleus is a very small and length is very large, and that's why what needed? Compaction needed, packaging needed, because in this has to inserted, this has to kept inside in the nucleus. We have seen already DNA inside the nucleus form, and the size of the nucleus is 10 to the minus 6 meter and length is 2.2 meter. So they need compaction, they need packaging. Okay, if we are seeing another that we have seen this one eukaryotic DNA, if we are seeing prokaryotes, so the number of base pair, just for example, number of base pair in E. coli. E. coli is what? Prokaryotes. It is bacteria. Okay. So how much base pair we have seen in E. coli? This is 4.6 into 10 to the power 6. So equal to 4.6 into 10 to the power 6. Okay. So this is the base pair in E. coli. And if we are taking their length, so length of DNA in E. coli. So what will come? 4.6 into 10 to the power 6 into 0.34 into 10 to the power minus 9 meter. Okay. So if we are seeing this one, so if we are minus 9 10 to the 6, so it will come what? Just for here we will see that 10 to the power 6 into 10 to the power minus 9. So it will come 10 to the power minus 3. And minus 3 what we can write? Milli. So if we are multiplying this 4.6 into 0.34, so it will come what? 1.36. So it is 1.36, and here it will come what? Milli and meter. So mm. Now you can see that length of DNA in E. coli is 1.36 mm. This is also bigger compared to typical nucleus size. So they need what? So this long size DNA accommodated in small area nucleus only through packaging or compaction. So what I told, long sized DNA accommodated accommodated in small area nucleus only possible by packaging or compaction. Okay, how it will be possible? We have to compact that one, or we have to package that one in the scientific manner. Okay, so that is needed packaging of DNA helix. Now we will see DNA packaging in prokaryotes, then we will see DNA packaging in eukaryotes. Now we know that in prokaryotes do not have defined nucleus. Okay, so come on that DNA packaging in prokaryotes, then we will come on DNA packaging in eukaryotes. Now, DNA packaging in prokaryotes. What we know, prokaryotic cell don't have well-defined nucleus. So what I told, prokaryotic cells do not have well-defined nucleus. Okay, they don't have well-defined nucleus. So what happened? Already we know double-stranded circular DNA is found in cytoplasm in a supercoiled state. So double-stranded circular DNA is found in cytoplasm in supercoiled stage. This term you have to remember. Supercoiled stage. Okay. Supercoiling occurring in the what? Cytoplasm in DNA packaging in prokaryotes. Okay. Now, this supercoiled structure is called nucleoid or genophore. So, this supercoiled structure is known as genophore or nucleoid. These terms you have to remember. Okay, supercoiled structure is known as what? Genophore or nucleoid. Okay, now the coiling is maintained because coiling always has to done because the size of nucleus is very small. So the coiling of DNA is maintained by positively charged non-histone protein that is known as polyamines. What I told, the coiling of DNA is maintained by positively charged positively charged non-histone proteins called polyamines. Okay, they are called what? Polyamines. amines. Okay, so what we have seen, this you have to remember also, that coiling of DNA, how this is maintained by positively charged non-histone, they are basic proteins. Okay, positive charge non-histone basic proteins, that is called polyamines. So that we have seen in DNA packaging in prokaryotes. Now we have to see how this DNA packaging in eukaryotes takes place. So come on that. Now DNA packaging in eukaryotes. What we know? Eukaryotic cell are having what? They are well defined nucleus. So the organization is much more complex, because inside nucleus we have to compact that one, and in prokaryotes cytoplasm were there. So in the cytoplasm that was easier than eukaryotic. But in eukaryotic, what is that? In eukaryotes, organization is much more complex, and that is carried out by set of positively charged basic proteins, that is known as histones. So what I told, in eukaryotes, the packaging of DNA in well defined nucleus is much more compact and complex, much more compact and complex than prokaryotes. Okay, then prokaryotes, it is having what? Much more complex. Now, it is carried out. How it is possible? It is carried out by a set of positively charged basic proteins, basic histone proteins, basic histone proteins. Okay, here what we have to see, basic histone, that was containing what? Non-histone proteins. So here basic histone proteins, or basic proteins, that is known as histone. Now these histones are rich in certain amino acid, that is lysine and arginine. What I told, histone proteins are rich in amino acid, lysine and arginine. Okay, this arginine and lysine amino acid rich present in histone proteins, helping in packaging of DNA in eukaryotes. Okay, now here what happens, these histone proteins generally having five types. What are the proteins are there? So there are five types of histone proteins are there. There are five types of histone proteins available proteins available as. Now what are the types are there? One is H1, one is as H1, H2A, H2B, H3 and H4. Okay, H1, okay, H2A, H2B, H3 and H4. Now this one H2A, H2B, H3 and H4. This double unit makes double means 2 into 4, this 4, so they are making octamer, and this H1 used to connect DNA, that is negatively charged particle. Okay, so H1 is not the part of packaging, but they are the linker, acts as what? Linker particle of DNA with octamer protein. So what I told, these 5 types of histone proteins. Now what I told, they are forming octamer. Okay, so these 4, H2A, H2B, H3 and H4. So 2 molecules of histone proteins H2A, H2B, H3 and H4 forms histone octamer. They are forming what? Histone octamer. So 2 into 4, that's why 2 into 4 is equal to 8. So each molecule, 2 molecule of histone proteins, they are forming histone octamer. Okay. Now these complete structure known as a new body. So these complete histone octamer having H2A, H2B, H3 and H4, now known as NU body. Okay, this also you have to remember, that complete histone octamer having H2A, H2B, H3 and H4, now known as what? NU body. Okay, now this NU body covered with negatively charged DNA. Okay, so what happens? The negatively charged DNA is wrapped along NU body. Okay, they are wrapping. This is what? Negatively charged, and histone octamer is what? Positively charged. So their negatively charged DNA is wrapped along NU body, or what we can say, NU body, or positively charged histone octamer now forms a structure known as nucleosome. Now understand what I told? This DNA wrapping along a new body or positively charged histone octamer, now it is called what? Nucleosome. Okay. Now if we are telling what are the situations that are coming, so we have to see that. Just for example, this is what? Two molecule H2A, H2B, H3, H4. Two molecule will come. Okay, so this is what? This is core of histone molecules. This is histone molecule. Octamer we also can say. Okay, now here what happens? Wrapping of DNA. Wrapping of DNA occurs, and this is H1. This is H1 histone molecule. This blue colored structure, it is DNA. So this is negatively charged DNA. This is histone is positively charged. So they are making what? Compactly attached to each other. And this combined form is known as nucleosome. So this is known as nucleosome. Now, what happens? How much they are forming nucleosome? So about 200 base pair of DNA is wrapped over a new body to complete about, this is what this turn is. This DNA turn is 1:3/4 turn. Okay, so this complete 1:3/4 turn makes one nucleosome. So what I told, we have to see that. Now what I told, the negatively charged DNA is wrapped over where? Around positively charged histone. Okay, so what we can say, the negatively charged DNA is wrapped over positively charged histone octamer, or what we can say, Nu body. Okay, now then here what we have, we already told about 200 base pair of DNA is wrapped over. How much? This line is very important. About 200 base pair of DNA is wrapped over Nu body. Okay, this is very important. Many numericals will come on this basis. So about 200 base pair of DNA is wrapped over where? Nu body to complete. How much turn is there? To complete 1:3/4 turns, and this forms a structure. This is forming a structure known as nucleosome. Now, so this is forming what? Nucleosome. Okay. Now, what we have seen, that the DNA present between two adjacent nucleosome is called linker DNA. Okay, what I told, the DNA, okay, the DNA present between two nucleosomes. The DNA present between two nucleosomes is called linker DNA. Now, this linker DNA, okay, with that this linker DNA that having base pair what? So with this linker DNA with about base pair 80. Okay, so with about how much? 80 base pair, and that is attached to H1 protein. This is attached to H1 protein. Okay, now one question already came that which protein is not the part of histone octamer. So which will be that? This one, H1 protein. Why? Because it acts as what? Connecting two nucleosomes. So it acts as linker DNA. Okay. Now, this nucleosome constitute repeating units because many nucleosomes are there, and that is called chromatin. So what I told, the nucleosome constitutes repeating units, and now it is called chromatin. One by one I am just want to say. Okay, so that is known as what? This repeating units is known as what? Chromatin. Okay, now if we are seeing this nucleosome chain, so that is gives a beads on string appearance. So nucleosome chain looks like beads on a string under electron microscope. Okay, this you have to remember. Under electron microscope, nucleosome looks like, so they are looks like what? They are looks like beads on a string. Now what is this beads on a string? Just for example, this is that are looking like this. This is a string, and in that string nucleosome are looking like beads. So if we are seeing through electron microscope, so that are looking like what? Beads on a string. Beads on a string. Where it will come? Under electron microscope. Okay. Now this nucleosome further coiled each other, and that is forming chromatin fiber or solenoid. So what I told, because many nucleosomes are there, these nucleosomes further coiled and form structure known as solenoid or chromatin fiber. Okay, they are known as what? Solenoid or chromatin fiber. Which one is showing? That nucleosome. Now, The chromatin fiber has a diameter of 30 nanometer. So the diameter now becomes 30 nanometer. This chromatin fiber having diameter how much? 30 nanometer. Okay, now this chromatin now behaving or differentiated into two regions. Okay, on the basis of staining. Okay, so now this chromatin behaving as two regions on the basis of staining of nucleus.